So yeah, we're about the sessions. So the aims will be basically to explain the principles of the techniques used for genomic DNA quality assessment, assessing for DNA quantification, assessing DNA purity, and assessing the integrity of the DNA. And applying these techniques to QC our DNA extracts, which, because we've already done the CTAB, but ostensibly you'll be doing everything from this point on till you put your your product on the sequencing one. And also we'll use the DNA quantification to quantify how much. So there's a step where you need to, when you put your samples onto the flow cell, if you have like one sample that's got a very high concentration of DNA, right? And you use 10 microliters of each, what's going to happen when you run is that you get loads of reads from that one sample that had like the really high concentration of DNA, and it will outcompete all the other ones, right? So you want to be able to get enough reads for all your samples, right? So you want to normalize your concentration. So for the one that has a high concentration, when you make up your solution, that's going to be taken through to sequencing, you're going to use less volume of DNA than for your samples where you got a lower concentration, right? You want to use more volume so that you get equal number of reads across all your different barcodes for your samples, right? So assessment of purity, it's done with the nano drop, and assessment of, you can also do quantity with the nano drop, but it's not, yeah, not for sequencing, because it's not, the absorption at 260. There are many other compounds that absorb light at 260. So it's not just the DNA. RNA also absorbs at 260. So yeah, it's not as good for sequencing. For sequencing, we prefer to use the qubits to quantify the concentration of the DNA, because then there's a specific binding agent that's binding the double-stranded DNA. So when you measure the fluorescence to determine the concentration, you know that it's bound to double-stranded DNA and not RNA or protein that may be in your solution. The TapeStation is like the top of the top as far as assessment of quality of your DNA is concerned. It will tell you what fragments, lengths of DNA are present in your sample, and it will also tell you their abundance in your sample, like their relative abundance, their concentration.
So it's a machine-based version of gel electrophoresis, and actually we found, because we have a TapeStation as part of our clinical trials, we found that actually the quantity calculations that it makes is always less than the qubit, and actually the qubits, nanograms per microliter, is far more accurate than the TapeStation. So we always qubit it to get the concentration, and then we TapeStation it to get the actual integrity of the DNA. Because I tried it a few times using what the TapeStation told me the concentration was, and I put way too much DNA in, and that doesn't really work very well. So qubit or the fluorescence-based assays, because there are other ones, are definitely the best for quantification.
Yeah. And then this will tell you basically if the fragment, for example, if you're doing a PCR, right, and you want to know the 300 base pair like fragments, are these the fragments that I'm getting? If you run a TapeStation, you know, like, okay, the fragments I'm getting are like the 400, yes. And, obviously to tell you whatever other fragments you've got in there as well.
Like for on a gel that
yes,
typically like it's like, oh, okay, it's around that 300.
Yes, exactly, because you have to run a ladder, right?
Within 2 to 4 base pairs, you can get resolution up to that amount.
But also it's really good for whole genomes as well, because it's got a high molecular weight one. It goes up to 60 kb, which with the nanopore, not necessarily. Even it's the same. It's really important that you have high molecular weight. The higher the weight, the longer your strands are. And for nanopore, that's really, really important. You can do this on gel electrophoresis. It's just going to take a bit longer, and you have to have a ladder that in theory should be able to reach the whole way to get it. We bought a special long ladder, so hopefully it works because I've not used it yet.
Yep. So when assessing for purity, obviously we don't want any contaminants that may affect our sequencing one, because obviously now sequencing one we have enzymes basically in the flow cell. Basically it's protein, right? So any impurities that may impede the functioning of the protein need to be eliminated from your sequence library once it's prepared.
Things can start to block the pores if you've got large things, and then I don't know, things like salts can affect the ion currents and things. So ideally just DNA. I think is what
so ideally we want as much DNA and as low impurities as possible. So with the nano drop, it can help you kind of do a baseline assessment of purity. So as we were saying before, the drawbacks are that even RNA like absorbs light at 260, which is the wavelength that the nano drop uses to assess the concentration of your DNA. But when you do comparisons of 260 to 280, the absorption at 260 and absorption at 280, you are able to kind of tell what the protein content is. If the reading you get is less than 1.8, the ratio is less than 1.8, it usually indicates higher protein content and other impurities. If it's greater than 2, you have a higher RNA contents when you do the 260 to 80 ratios, right? And then when you do the absorbance at 260 and 230, if it's above 2, it indicates the presence of contaminants in your solution. So, the box over there. So yeah, that's just a picture of the nano drop. So you put your like 1 microliter volume on that stage there, and then the light comes through the bottom and it measures the absorption. And
I just need to download the software to install it.
Sure.
Okay. Then we can connect to that.
It's not necessarily okay. When you this would need it to me.
Okay. All right. So that's purity that uses for the quantity and concentration of DNA. So it comes with the kit. The stock of working solution, and then it comes with 2 standards. And then basically dilute. If it came with the stock solution, you have to dilute down the stock solution. You add 197, well, between 1 and 10 microliters of your sample to between 190 to 199 microliters of your working solution. And then you measure the concentration that you get from there.
I should just point out that target is for the original kit. I haven't changed that. So the target now, if you wanted 400 nanograms in 9 microliters, which is what the highest input, so the kind of optimal input is, that actually works out to something like 44 nanograms per microliter. But with this new kit that we're going to use, it can be anywhere down to 5 nanograms per microliter, so 50 nanograms. So anywhere between 5 and 44 is kind of optimal. Obviously above that's great. But anywhere below 5, and you would probably not want to run that because it wouldn't be enough.
So these are just the list of the different kits you can get for the qubits. We're currently using the high sensitivity kits to measure the quantity of DNA in our samples. So this is just telling you about how you make up the solution. So for the standards, you get 190 of your working solution and add 10 microliters of the standards. There are two standards, S1 and S2. One of them has a high concentration of DNA, the other one has a low concentration of DNA. And then the idea is that all your samples will fall within that range. And then for each one of your samples, what will we be using? We're using 1 microliter.
So you can use anywhere between 1 and 10 microliters.
Yeah.
But as we were saying, if you're doing CTAB, you're probably going to be eluting into a really small volume.
Yeah.
So I think 1, 2-ish.
Yeah.
I think some
1 or
people
2
say 1 isn't
microliters.
accurate enough. We tend to do 1, and that seems to be working fine.
Yeah.
If you want to do more, then that's fine.
Yeah.
But obviously, 10 microliters is an awful lot, especially if you're eluting into 20. So
Exactly.
yeah, 1 to 2, 1 to 3 is
Yeah.
it's probably fine.
Yeah, we were using 3 microliters before, just because at 1, sometimes you get, like, if you measure the same sample twice, sometimes you get a bit of variance if you use 1 microliter. So yeah, and that's just the dilution practice. So basically the qubit will tell you the final concentration. So it's tell you the concentration in the tube and the final concentration of the sample that you took it from. And you will tell the qubit exactly how much of the sample you're adding, whether it's 1 microliters or 2 microliters. So then with those calculations, it will tell you what the final concentration of the sample that you got was. So yeah, this is basically just outlining the steps that I've already spoken about. So these will be your standards, 10 microliters into each. These will be your samples. So add the working solution there, depending on how much sample you're adding. So if you're adding like 1 microliter, you're using 199 microliters of working solution, because the final volume is 200 microliters. And then you start with your standards. You start with on standard 1, measure, standard 2, you'll measure the concentration, then get the curve, and then add your samples and get that curve and to tell you what the concentration is.
You can tell the qubit how much DNA you put in, and it will then calculate based on that what your actual concentration is.
Yeah.
So if you put 10 in, obviously you do a dilution as well. So it will still know.
So as far as integrity goes, we're looking for appropriate molecular weights. And so we can use the gel electrophoresis to do this. So principle of gel electrophoresis, fairly basic. You're moving a fragment of DNA through the agarose matrix, right, under an electric current. So obviously the larger strands migrate slowly, the smaller strands migrate rapidly. Right? And then you run a ladder with it, and then you compare the ladder, since you know what size fragments are in your ladder, you compare them to how your sample was run to determine whether you've got the correct size fragments. Yeah. So this is just an example. So this would be the ladder on your far left. And then you have your samples. And then, ideally, what you want to see, if you did everything excellently, is like one, bright band. Especially if you need it as a PCR, right? One bright band products, right? Since we're doing culture and then we're extracting, you have various fragments, but what you want to see is that the higher molecular weight fragments are very well represented, right? So obviously, your agarose gel that you make would depend, the concentration would depend on, how long you want to run the gel how much resolution you want. So in this case, it's 0.7%, so 0.7 grams in 100 mils of water. And obviously the lithium bromide is used to bind to the DNA, so you can see it when you are using your gel documentation machine.
That's a very bad gel, by the way.
This one?
Don't aspire
Yes,
to
it's
that.
like a smear, right? More like
But don't aspire to that.
yeah. It's more a smear than discrete bands. So that's just like the equipment that you use when you're running a gel, right? You've got your tray, your electric leads, your power supply, obviously, your gel tank, where you put your buffer in and you submerge your gel. So for like larger fragments of DNA and for like resolution of those fragments, sometimes we use pulse gel electrophoresis. I never had I did this once when I was doing my master's degree, and I've never done pulse gel electrophoresis again. So
You should get away with just.
yeah, we can get away with just running a conventional gel.
Might just fall off the end, but hopefully we'll be fine.
Exactly. So that should give you enough resolution. So like she was relaying earlier, you want discrete bands and not this like use like smear, right? You want more discrete bands, like you can see on the right hand side. So like discrete bands with
If you've got that smear, it means you've got lots of smaller chunks of DNA, and that tends to, for some reason, despite it being long read technology, preferentially seems to sequence the small bits you don't want.
Yeah.
You could potentially do a bead wash to get rid of those small bits, but to be honest, for that amount of effort, you're probably better to just redo the extraction, I think.
exactly.
A is definitely
Because with the bead wash, you tend to lose some DNA when you wash with the so you might just want to re-extract. So yes, the TapeStation. So with the TapeStation, big machine, then you have like this little small tape that has dots where you're supposed to add your samples, right? And then it's basically just like rapid gel, like running a rapid gel. It's very well. It's not an exact sample, but it's gel there. It's very, very quick. You can do. So each tape is something like it's got 16 spaces. So you have one for a ladder, so you can run 50 gel sort of blades, and it takes about 40 minutes. So it depends what you're doing. If it's large whole genome stuff, then it takes longer than if you're doing a smaller gel. But yeah, it's very handy to get, but it's also quite expensive. Exactly. And the bioanalyzer is kind of a precursor to that. So it provides you with a sort of what's the peak, just how big your DNA is, whereas the TapeStation actually provides you with the gel picture itself. So yeah. Just so this is basically how it looks. Comes like kids, they'll be like the tapes themselves, the boxes, and then reactants. If you're doing a lot of sequencing, or it's kind of something that's going to be really important, I think investing in something like a TapeStation really be quite easy. Just get a small version. Yeah. Or a large version. I mean, especially if you're using Illumina, it's a must, because with the Illumina, you don't want the primer dimers, which are like the smaller bits of DNA that then bind together, that then amplified so much more. Even if you've got a small amount, it affects your RAM. I think Agilent is generally available in Africa, right? Agilent is generally available here. Because I know that sometimes it's hard to get the things.
There's also now from Taiwan. It's called Biotics.
Okay.
Fusion 100.
Yeah.
So they have sort of like particles where that capillary.
Yes, it's right.
So you can just slow that in. And I think in terms of the reagents and those, I think much cheaper.
Okay. Yeah.
So each cartridge is about there are 2 cartridges inside, and each one can do 200 runs.
Oh wow.
Yeah, and I think it's under $100.
Oh, brilliant.
Yeah, so that's $400 for $100 a sample. That's very cheap.
Yes.
And then it takes about 4 minutes per sample, and you can also take plates and
In the machine itself,
29,000.
29,000.
And the TapeStation.
Okay.
So that's also, that's what we want.
Yeah, okay.
So that's also something which one can consider.
Yeah, other models are available.
And
It's the same
it's
Yeah,
the same principle, yeah.
it's, a robot gel machine.
So that's like a picture of the tape. So that's what it looks like. And I think when it says D1000, that's the ladder, right? That's
You
the
have-
size ladder that you can
Yeah,
buy.
the different kits, so
Yeah.
you will get, the gels are, so you get different tapes for different kits. And you get obviously different reagent kits, so if you're doing Illumina, you can buy really small sized kits, or sort of say we use the gDNA ones. So I imagine because they run roughly the same, I imagine maybe the gel is thicker potentially in the smaller one, so you get better resolution. I don't know exactly how it works, but yeah, you have different kits and different tapes for different
Yeah.
processes, so
So
yeah.
there where it says D1000, that's the largest like fragment it has in its ladder. So basically that's.
But now with they changed the chemistry slightly, not slightly, they changed the chemistry group. So in terms of your input, it's now pretty much the same level that often, because if this is your fragment and it's so small, when you do the cut and tag, it cuts it and it's less than 500 base pairs. It means that when you do 150, they sequence into each other.
Yes, exactly. Yes.
So they need to be, they need to be as big enough a
Yes.
fragment in terms of, so you're also looking for the ones which are pairing there, then as it is cutting, it's giving uniform fragments to give you more paired in driver.
Yeah, at various stages, it's important to identify size and quality.
Quality,
yes.
Quality of your DNA. So throughout this, before you even put your samples in to an Illumina and nanopore, you want to see if you've got nice DNA in the first place. But then afterwards, if you're pooling your samples, you might also want to check, certainly with Illumina, you need to check what sizes you've got. Nanopore, it's less important to
So
do
that's
it.
what the Terra XT now.
Yeah,
So it's now DNA prep.
Yeah.
One is not need to do the individual fragment analysis of like the way it was. So it's much more focused.
yeah.
And it was very thin.
Yeah.
It wasn't forgiving before. Much more forgiving.
So yeah, if you are doing sequencing on a fairly big scale, then it's definitely worth automating this process if you can.
So these are examples of, so on the right is like the picture you get from the TapeStation. It will show you like what your lanes were, and there is your ladder. On the far left, on that one is the ladder, and then as you can see like a smear in the middle there. And then it will give you on the computer, you get the readings that are on this side here. So at different points where you see those bands, you get a peak corresponding to that molecular weight.
What TapeStation also does is give you a DIN. Oh, so you can see it here at the bottom. So those DINs. So DIN is DNA integrity number.
Yes.
And the lower the number, the worse your DNA is basically. And I'm not sure if you can properly read it, but you can see A is supposed to be rubbish DNA. You can see it's smeared. You can see that the peak isn't a peak, it's more of a hump. And the score is 2.9. And over there on the other side, B is really good, nice, happy DNA. And you can see the DNA integrity score is at 7.7.
7.7.
Yeah, so the higher the number, basically, the better your integrity of your DNA is. So you're aiming for, and I think the new versions of the analysis software actually give you color coded DINs. So it gives you red if it's sad, yellow if it's kind of medium, and then green if it's like good. It gives an awful lot of kind of analysis, this program. I don't know if the Taiwanese one does the same. Does it give you DINs? Does it give the DNA integrity number
Score.
on the?
The bioptics.
Yeah.
So
Can you just show, do you have an example from?
Yeah.
yeah.
So down at the bottom,
So
those blue
bottom
squares.
right, there where it says 7.7.
Yeah, it does.
Yeah.
Oh, it does? Okay.
Yeah,
Alright.
it gives me some form of, but typically I would still look at the curve itself to see, yeah, but it does, yeah. It does.
The better your curve, the higher the score in theory.
Yeah.
So basically, it's just a slide telling us like what we're aiming for roughly. So like we said, with the new kits, well, we can get a lower concentration of DNA. We can get like 5 nanograms, and we should still get a good sequence. From that, for molecular weight, we're looking at larger fragments, like we said, where we'll get more coverage, the larger the fragments that we get.
This
We're
kit, I
looking
think,
for, sorry, go ahead.
I was going to say, this kit specifies the minimum molecular weight input should be 30 kb. So if you have less than that, then it's not optimal and you won't get as much. Generally, it doesn't sequence as well. The higher the molecular weight, generally, the better. Although, as you will see when I do my presentation on YTB and pain in the bum, actually sometimes shorter is better.
So this is just looking at calculating the molarity of your DNA. This is when we're pooling the DNA and we're going to the steps, the later steps of putting it onto the gel. There'll be a stage where we barcode and then we'll pool all the DNA into one tube. And then that's the tube that will be loaded onto the sequencer. So we're going to take you through all these, all the calculations that you need to do when preparing your when doing your library prep. I'm sure this thing will be this calculation will be revision for those who did pure sciences. Yes. Yeah. So that's the last slide. Any questions?
The kit itself, which should be used for anything. So it specifies that this new kit, the new version of the kit that we're going to be using, specifies you should have at least 30 kb as your average molecular weight for all of your samples, and you should have somewhere between 50 and 200 nanograms of DNA. Although, as I say, I've been speaking to our nanopore team that we work with, and they actually said that putting in 400 nanograms, which is what the original kit suggested, is actually better because nanopore works on a basis that if you don't have very much DNA, the fragments will go through, but then it can take a while for another one to find its way to the core. If there's more DNA, it's like a combine of samples that will go through, and it makes it more efficient. And so you don't want to overload it, but actually you do want to put quite a lot in to make sure that that sort of flow through the core is going well. And also, if you have lots of small bits, that also stops that kind of efficiency. And so having this long, and this is, I think, why they said 30 kb is a minimum. If you put less than that in, it goes through really rapidly. Like, I think it does, I don't know how many bases it does a second, but it's really, really fast. And so that DNA will go straight through very, very quickly. And so if you have a long piece, then it's going to be able to like join up with another one. But if you have short bits, then it's got to find its own way to the core, is how I understand it. So, but as I say, with TB, because you have so many repeat regions, what I think has been happening, because it seems to be less efficient, as I've said, is that when your DNA goes through and you get your one strand going through and the other bit floating off, I think possibly that other bit is forming sort of primer dimer balls on itself.
Yes.
And possibly clogging the pores. So we've tried it, we've cut it down 60 kb, 30 kb, 20 and 10, I think. And actually, we found that rather than having high molecular weight that usually you would want, actually somewhere around 30 or even 20 kind of works better. But then there's a trade-off between you're getting half the amount of information because your read is half as long. So it's a bit of a trade-off again.